Dear students, our today's topic is regulation of gene expression. You have already studied how is a gene expressed. You know, gene is a segment of DNA. This DNA is first transcribed to an RNA, and this mRNA is then translated to a protein, and likewise producing number of proteins, our characters are expressed. Then our question is, are these proteins produced all the time? Is the synthesis constitutive? Is there any regulation by anything? Okay, how is the protein synthesis regulated? Is it at the time? I mean, if we need a protein, is it synthesized at the time? Or is it synthesized all the time? That is what we are going to discuss. That is regulation of gene expression. See, gene expression regulation can take place at 4 levels. That means the first level in the transcriptional level. Gene expression can be regulated in the transcriptional level. Second, in the processing level. Third, in the transport level. And fourth, in the translation level. Translational level. What do you mean by this? What do you mean if I say the gene expression is regulated in transcriptional level? That means at the transcription itself, it is controlled whether it should be done or not, if it's needy or not. Okay, that means if the particular protein is required, then only that is transcribed. Otherwise, the transcription is blocked. Okay, so this is transcriptional level regulation. Whereas in the second case, processing level regulation means you have studied many processings, especially splicing. That means transcription occurs, but splicing occurs only when it is needed. Okay, the protein, the DNA is transcribed to RNA. It is present inside the nucleus. It is not transported and it is not spliced. When it is required, that time only it is spliced. So, in this level, it is regulated. In the third case, okay, these two processes might have done, but it is transported out of the nucleus only when it is required. Therefore, we can say it is transport level regulation. Okay, these two, transcription is over, processing is over, and all the mRNAs are processed and kept in the nucleus. And whenever it is needed, it is transported out of the nucleus. Then we call it as transport level, it is regulated. And last case is regulation in translational level. That means transcription is over, processing is over, it is transported to cytoplasm, but translation occurs if it is needed. In the translation process, there is the control. What if the protein is required in high amount? More translated. If it is required in less amount, only less translated. If it is required it is not required means not translated. That means translational level regulation. Is it okay? Today we are discussing a transcriptional level regulation. Before moving to that, okay, this all regulation is taking place inside the body. Internal regulation we can say. Okay, what controls the gene expression is our processing. Our own body processing. Whereas in some cases, external factors also, along with the metabolic factors, external conditions can also regulate gene expression. Okay, so for an example, where external factors regulate gene expression, we give the example of lactose. Okay, in the case of bacterium Escherichia coli, okay, the most used experimental material. In the bacteria Escherichia coli, beta-galactosidase enzyme is required for the metabolism of lactose. And this enzyme split or break down lactose to glucose and the galactose. You know that, okay? It's a disaccharide made up of glucose and galactose. That means if Escherichia coli bacterium gets lactose in the medium as food, okay, if this bacterium gets lactose as the nutrient, this requires the breakdown of lactose. Okay, if it's getting lactose as the food or nutrient, that should be broken. Then it requires this enzyme. Only the requirement is only to break down lactose to glucose and galactose. Suppose this bacterium is not getting lactose from the medium. Okay, it is growing in a medium where there is no lactose. That means then this enzyme is not required for the bacteria. Okay, so such a case, E. coli produces this enzyme only if there is lactose. That means the presence or absence of lactose in the surrounding, that means in the environment, is controlling what this enzyme synthesis. This is a protein. Okay, almost all enzymes are made up of protein. So understand what is controlling the gene expression here. Here the gene expression is, which gene expression? Gene for beta galactosidase enzyme. That is expressed only when there is lactose in the environment. Okay, so what is controlling the gene expression here? Environmental factor. If lactose is present as an environmental substance, nutrient, then only the gene for this is expressed. So understand, not only our body, internal processings, the environmental factors also can control gene expression. For that one example you have learned, this is the best studied material, and so many other examples are also there. Okay, these things are clear, I think. Okay, how is gene expression regulated? And we are moving to the first case, transcriptional level regulation. This is the most common type, a regulation at the first step itself. Okay, that means a cell is not producing unnecessary RNAs. When it is needed only, it is producing RNAs. Okay, that means transcription is only if it is needed. So transcriptional level regulation. In most of the prokaryotes, prokaryotes are the best studied material for the gene expression regulation and all such kind of experiments. So we are discussing transcriptional level regulation with the help of some examples from prokaryotes. Transcriptional level regulation in prokaryotes. Okay. See, you have studied in prokaryotes, its structural genus what? Polycistronic. Prokaryotes polycistronic, eukaryotes monocistronic you have studied. In order to remember that, you remember PP. Prokaryotes polycistronic. Okay. So, see, in prokaryotes, the structural genus polycistronic. That means, what was your concept? There is a common promoter and terminator for what? Menicistron. Okay, that means the structural genus polycistronic. It is here many genes are there. We will discuss with an example later. So, anyway, in prokaryotes, its structural genus polycistronic. Then understand for its regulation, gene expression regulation. Suppose here I am giving an example. Okay, so if this is a transcription unit, what is the first sequence called? What is the sequence called? This region is promoter, this region is terminator, this region we call what? Structural gene. Okay, I think you remember it. So, promoter, terminator, and in between, what is flanked in between is structural gene. And in prokaryotes, here it is having polycistronic means one transcription unit consists of many genes. We will discuss it. Okay, suppose this is a case of Escherichia coli. Now, just now we discussed lactose operon. What is an operon? I will make you clear. Okay. Suppose this is the case of Escherichia coli bacteria, a segment of DNA, transcription unit. And according to operon concept, okay, the gene expression regulation is carried out by some sequences in the transcription unit itself. That means if there are three, suppose Z, Y, A. These are three genes of the transcription unit. If there are some three genes in the transcription unit, its expression is controlled by certain sequences present here in the transcription unit itself. That is what is the concept, operon concept. That means an operon is a DNA segment which consists of structural gene portions and also its control portion. So in order to control the expression of the structural genes, in order to control this expression, there is a control region. Okay? What are the parts of the control region? See, in the lac operon, the example we are going to discuss, in the lac operon, lactose operon, within the promoter, promoter sequences from here to here. Okay, suppose from here to here is promoter sequence. Inside the promoter itself, from here to here it is promoter. So within the promoter itself, there is a region called regulator gene. We are naming it as regulator gene. Okay. So one part of the operon is regulator gene. Then after the promoter, here there is another region called operator gene or operator sequence. So understand what are the essential parts of this operon. This operon consists of a functional structural gene portion. These are genes to be expressed. It consists of a terminator, it consists of a promoter, and additionally it consists of a regulator gene and an operator gene. Is it okay? So you define what is an operon. What is an operon? An operon is a DNA segment with a promoter, regulator, operator, structural gene and terminator. That means it is a transcription unit. What else you have studied additionally is what? Regulator gene and operator gene. Okay. Then with the help of this lac operon example, you are going to study the gene expression in detail. Which are the genes to be expressed in this operon? Z, Y, A are the structural genes to be expressed. So, since it's polycistronic, there is a common promoter and terminator for these three genes, three structural genes. That is the concept of polycistronic unit. Okay, Z, Y and A genes are controlled by a common segment. Okay, so this is the terminator. Then tell me, Z, okay, just study these names also. Z is a gene. Z is a gene which help in the production of beta-galactosidase enzyme. Z gene transcribes beta-galactosidase enzyme. Transcribe the RNA and then beta-galactosidase enzyme. Y gene is responsible for forming permease enzyme. Then A gene is responsible for forming trans-acetylase enzyme. These names you have to study anyway. Okay. Then this is just an example. You have to understand what is the role of promoter, what is the role of regulator, what is the role of operator. Role of promoter, terminator you have already studied. What is the role of promoter? It's a region where transcription begins. RNA polymerase enzyme can access this region only. Okay. Then, I think these parts are clear. Then we discuss the roles of this. Then lac operon. Okay. So, what is the role of I gene here? The I gene is the regulator gene. See, a regulator gene. Here it is named as I. In other operon, different name may be there. A regulator gene, here it is I. Okay. Then it is either producing activator protein it acts by two ways. Okay, or it is producing repressor protein. Here regulator is the sequence. Here the name is I. It stands for inhibitor. Okay, I, expansionist inhibitor in this particular example. We will discuss it. And any regulator gene is acting by forming either an activator or a repressor. If it produces an activator, it's a positive regulation because it enhances, it activates the transcription. If it is producing a repressor, it's negative regulation. It is blocking the transcription. We will discuss with an example. Okay, then the role of promoter, you know, it is to start, make the RNA polymerase attach to it, recognize it and start the process. And the role of operator, the regulator gene is acting, the product of the regulator gene. It was activator or repressor, its action site is operator. Okay, suppose if a repressor is produced, it is acting on the operator's region. So these are the functions of this region. If these basics are clear, we move on to the lac operon concept, which is produced by, given by Jacob and Monod. Francis Jacob and Jack Monod. Gene expression has transcription, translation, two main steps, and in between there is processing and transport. For all these four levels, gene expression can be regulated. In the transcriptional level, in the processing level, in the transport level, and also in the translation level. The best studied example, prokaryotes like E. coli, most of them show most of the protein synthesis is regulated in the transcriptional level. Okay, and when the in the transcriptional level, when it is regulated, there is the concept of what? A regulator gene. And it is by the regulator gene action, the process is regulated or controlled. The regulator gene is either producing an activator which positively regulates the process or producing a repressor which negatively controls the process, which blocks the process. Okay, and whatever it is, the regulator gene's product, whether there is an activator or repressor, its action is on the operator region, operator gene. So that is also an essential part of the control segment. So the concept of operon says that an operon is a segment of DNA which has a control section and a structural gene section. Okay, the structural gene, if it is prokaryotes, it is polycystronic. Here it has 3 genes. It varies. Okay, so if the control unit portion consists of promoter, then operator and regulator. So an operon can be defined as a segment of DNA with a promoter, regulator, operator, structural genes and a terminator. The transcription unit itself. Okay, you have studied 2 more additional things in that. I think it's okay. Then we move on to the example, lac operon. It is expanded as lactose operon. It is one example for operon. There are many other examples like val operon, valin operon, tryptophan operon, type. Okay, ala operon. Okay, like operons are there, many. So, example for an operon, lac operon. Okay, and before moving to the operon, I have given you an idea. See, if lactose is present in the surrounding medium, then only the bacterium E. coli requires the transcription in order to metabolize lactose. Okay, if it's not, lactose is not available means the synthesis is not necessary, it can be blocked. So understand 2 cases. If lactose is absent in the medium, the transcription is not necessary. Why? This enzyme production is not necessary because lactose is not available, no need its breakdown. Okay, so if lactose is absent in the medium, no transcription is occurred. How it is, we will discuss. If lactose is present in the medium, transcription must occur. These enzymes are required for the lactose metabolism. Bacterium is getting lactose, it should metabolize that lactose. It should break down the lactose into glucose and galactose. For that, these 3 enzymes are required. So, transcription must be there when lactose is present in the medium. It should not be there when lactose is absent in the medium. That is what we are going to discuss. Okay? different parts of lac operon. Lac operon has a promoter. Within the promoter, there is a regulator gene. The regulator gene is named as I. Okay? And after the promoter, there is operator gene O. Three structural genes Z, Y and A. Okay? These are the parts of what? Lac operon. The terminator also is there. Okay? Then, you know, RNA polymerase enzyme comes to bind promoter. Okay? That idea you have. Then all the time, understand, constitutively, all the time, this gene, I gene expresses to produce an mRNA, and that mRNA is translated to a protein which is a repressor. So it shows what type of regulation? Negative regulation. Here it is not, the regulator is not producing activator, it is producing a repressor. All the time the repressor is produced. Okay? Whether it is lactose present or absent, all the time this gene expresses, produces an mRNA, then produces a repressor protein. So constitutively expressed gene. Okay? Then understand, if lactose is absent in the medium, this repressor molecule goes to bind to the operator region. That's what I have shown here. Okay, to block the operator sequence, there is a block. Then imagine our enzyme, RNA polymerase enzyme has reached here. It is opening the helix and moving to here. Here is a block. It cannot proceed. The RNA polymerase enzyme cannot proceed after this because there is a block by repressor protein. Did you understand this? That means there is no transcription of Z, Y and A gene. These genes are not transcribed because the repressor protein binds at the operator so that this enzyme cannot move ahead. Okay? Cannot move ahead and it cannot do the transcription. Is it okay? So what is the block here? The repressor protein is the block here. So you know it negatively affected. It will block the process. Okay. Then if lactose is present in the medium, what is the process? If lactose is present in the medium, understand repressor is always produced, but this lactose goes to bind to the repressor. To the active site of the repressor protein, lactose goes and fit. Then what happened? It inactivated the repressor. This active site is not free to bind here. That means this repressor is inactivated so that it cannot block operator. There is no block on the operator. Then what happens? RNA polymerase comes, it opens the helix, goes on transcribing, okay? It reaches this site. It can easily move ahead. All the three genes are transcribed, okay, to form a what? mRNA. This first to form an mRNA and this is translated to three enzymes. From this, this translated to beta-galactosidase enzyme. Y the region, this portion of RNA is translated to permease region. A is translated to trans-acetylase region. Three mRNAs, okay, then forming three different enzymes. Why these three different enzymes required? All the three enzymes are required for the lactose metabolism. For lactose breakdown, lactose to glucose and galactose, this is required. Beta-galactosidase is required. Permease is required for the increasing the permeability to absorb. Okay, after the breakdown, this gives some products. Its absorption to the cells become easy by this enzyme. Permease enzyme increases membrane permeability for the fine absorption. Then trans-acetylase enzyme also help in the transfer. Okay, so that is the function of the three enzymes here. All the three enzymes are required for the effective metabolism of lactose. So that is required to be produced only when lactose is present in the medium. Okay, if lactose is present in the medium, lactose itself acts as the inducer molecule. Okay, inducer molecule. Lactose is the inducer molecule here. Lactose can block the repressor. Okay, then there is no block on the operator. These three genes are transcribed and then translated to This 3 enzyme. This is required for the breakdown. These two are required for the transport, effective transport. Okay, that's about the action. See, other than lactose, allolactose also can be an inducer for the same action. Allolactose. Okay. Then one more thing. There are two more terms to study from this. One is positive regulation. Positive regulation. See, this lac operon is said to be under positive regulation when it is controlled by the inducer molecule. In connection with inducer molecule, when we speak, lac operon is showing positive regulation. What do you mean? If inducer molecule is present, lac operon functions. If inducer molecule is absent, lac operon stops functioning. So, in terms of inducer molecule, what is the inducer molecule here? Lactose. In terms of lactose or inducer molecule, the lac operon shows positive regulation. Some other concepts are there that we will study in higher class. Okay. Then, in terms of the repressor, lac operon is under negative regulation. What do you mean? In terms of repressor, when we speak, that means presence of repressor is blocking, absence of repressor is switching on. Okay, so once again, the lac operon is under positive regulation in terms of the inducer. That means if inducer is present, lac operon is switched on. If absent, switched off. Then in terms of repressor, when we speak, it is under negative regulation. That means if repressor is present, it is switched off. If repressor is absent, it is switched on. Okay, it is never absent, but it is inactivated. I think this topic is clear. That is about the lac operon. This is the most simple operon to study. There are other operons with more number of structural genes and it is entirely different. The action is entirely different compared to this. And this is the most simplified explanation of a lac operon. Any more doubt means we will discuss it at the end. Okay? Thank you.